Greetings. I would like to welcome everyone today to the Health Informatics Lecture Series for the spring 2023. The purpose of the lecture series is to bring experts in the field of information, and we usually select individuals that they're dealing with the area of informatics and its application to health and healthcare in its broadest sense. First of all, I would like to say that I am Anna Cleveland. I am a Regents Professor. I am the Sarah Lowe Endowed Professor, and most applicable to this, it is that I am the Director of the Health Informatics Program. Usually at the beginning of our lectures, Dean Kenchik, the Dean of the College of Information, give his welcome, but this time he's in a Dean's meeting, and as soon as he's finished that, he will join us, and he will give the closing remarks at this time. Before I introduce our wonderful speaker, I would like to introduce Dr. Jingpeng Chen, who is the chair of the Department of Information Science. Dr. Chen, would you like to say a few words?
Sure, Dr. Cleveland, thank you so much. Thank you. Thank everyone for coming to this wonderful event that Dr. Cleveland and the Health Informatics Program has been organizing for I think a few years. And today is our great pleasure that we have Dr. Lu joining us. Dr. Lu, thank you and welcome to be here online to talk to our students. And it's impressive that we have so many participants today. I'm really impressed. So Dr. Cleveland and Dr. Lu, thank you all for this wonderful event. I'm so looking forward to Dr. Lu's presentation. Back to you, Dr. Cleveland.
Thank you, Dr. Chen. Also, I would like to acknowledge that Dr. Yunfei Du, it is in the audience. He's the Associate Dean for Academics for the College of Information. So Dr. Du, thank you for joining us today.
Thank you for the opportunity. Thank you, Dr. Lu. Welcome.
Thank you. Thank you. So at this time, I would like to say that it's a great honor to introduce the speaker for today. But before that, I hope you allow me to say just a personal note of gratitude to the National Library of Medicine. Before we started the lecture, I did mention it to Dr. Lu that I'm a product of the National Library of Medicine and the National Institute of Health. I would not be here without the wonderful support that they gave me for my master's and my doctoral program. They gave me the vision of what was all the role of health information, especially for patient care, and what role do we have for the clinician and the healthcare providers. So I'm expressing my thanks to you, Dr. Lu, because you are part of NLM. So our speaker for today is Dr. Zhiyong Lu. He's a senior investigator for the computational biology branch at the National Center for Biotechnology Information. He serves as the deputy director for literature search and is a member of the extramural research program at the National Library of Medicine at the National Institute of Health. Today, he will present the work on developing large-scale machine learning-based tools for better understanding the scientific text in the biomedical literature. The topic of Dr. Lu's lecture, it is AI in medicine, from PubMed search, we say, to medical diagnosis. So Dr. Lu, we want to thank you for coming today, joining us, and share your expertise and your vision of where we're going in the future with all these machine learning applications that right now there's so much in the forefront of our society. Dr. Lu, the floor is yours.
Thank you so much. Thank you so much for the kind invitation, Dr. Cleveland, Dr. Chen, and Dr. Du. So it's my great pleasure to share some of our recent work with you all. So as introduced, I am a group leader at the National Library of Medicine Intramural Research Program. I supervise a group of 20 PhDs, and we are interested in using machine learning and AI for processing both biomedical text and biomedical images. So on a daily basis, we primarily work on biomedical literature. When access is allowed, we also work on patient electronic medical records, as well as clinical images. And in that regard, we mainly working in the field of ophthalmology and radiology, because we have great collaborators on the NIH campus. So using machine learning and AI, we work on a wide variety of different problems, ranging from improving information access for the biomedical literature to extracting and turning unstructured text into structured information and knowledge, to develop using AI and deep learning for autonomous disease diagnosis and prognosis. So that is basically the kind of, so I'll talk in my talk, I'll give you about 3 stories, one from each areas. So let's start and begin with the literature. So, I don't know the audience, how many of you are familiar with the PubMed? I assume you all know that because you're working in the field of health informatics. So PubMed is a database of biomedical literature, and today, and this number is already outdated, we have over 35 million articles in PubMed. And on an average day, we have about 2.5 million users. They're doing 3 million searches and 9 million page views. So even though AI and machine learning is a hot topic these days, but since a long time ago, we have been using AI and machine learning for improving PubMed search. And that's actually since day one, since I started at NCBI about 15, 16 years ago. So this slide just lists you a number of features or search assistants that are made available using computational approaches. And in fact, a couple of years ago, we even wrote an article to sort of explain how we make these features available behind the scenes. So if you're interested, you certainly are very welcome to read this paper in detail. But today, given the time, I can't go through all the list of features, but I do want to give you one to give you an idea how AI and machine learning is used in PubMed search. And that is something called a best match, which is sorting results by relevance. So if you are, if you use PubMed for your research, you probably, and have been using that for a few years, you probably are familiar with this interface, which is the previous version of PubMed. And at that time, all the results are basically sorted, can only be sorted by date order. In other words, you always see the most recent papers in the front at the top. So when we introduced the best match, we added the most relevant papers at the top of regular search date sorted order results. So people often ask me, why do you, since day one, PubMed has been using date sorted order, and that seems to be worked fairly well in the last 10 years. So why do you, or 20 years, why do you want to change or making a different sorting order? So one thing we would address is that things have been changing because PubMed content has been growing very rapidly. Every year, we see more than 1 million papers entering an index by PubMed. And today, like I said, we have over 35 million papers. And every minute, that's about 2 to 3 new papers come into PubMed. So we've talked about maybe 5-10 minutes. And that means that there are already 20 papers that we didn't get a chance to look at. Now, of course, you will say, not all the papers are relevant to my research. And that's only very true. But think about something like the COVID, like the past pandemic. When we started looking at the papers in PubMed about COVID-19, we started about only a few hundred papers. But that number has changed so quickly now that we have over 250,000 papers in PubMed about COVID-19. Okay, and also to mention that term COVID-19 was not coined until summer 2020. So before that term was used in papers, there are a lot of different ways that COVID-19 was described. Therefore, we also put together a database just for COVID papers. That's called a LitCovid. So not only the content, the sheer volume of PubMed makes it very challenging for researchers to use and to retrieve papers, relevant papers, but also user behavior is changing. Okay, ask yourself, when is the last time you actually go to Google second page to find something? Now, it's certainly still possible, but I guess that's very rare. And you may say that, you know, Google is Google and PubMed is PubMed, but actually PubMed users like me, like you, probably also using Google for our other search needs. So when we look at the PubMed, it's actually very similar. You know, 20 is a magic number here because, as you can see, 80% of the user clicks happens to the first 20 results, because in the past, PubMed returns 20 results by default in its first page. Okay, so therefore, that really motivated me and one of my former postdocs, who's now working in a search industry, to come up to trying to optimize the search results in the first page, because that's where most of the user action is about. So we wanted to provide, offer users an alternative to the reverse or sort of order to ranking the papers. How do we do that, right? So there are classical traditional way of doing that, just looking at the matches between the query and between the words in the document and trying to compute relatedness between the query words and the words in the document. So that's the traditional way of doing that, called BM25. But we sort of, especially Nicholas Furini, the postdoc here, and he decided to investigate a number of different methods, and he finally decided to let's try a learning to rank, a machine learning type of work for ranking the papers. Because using machine learning, we can actually not only using one ranking signals, we can actually including multiple different ranking signals to determine what is the best for our users. Okay, but if you're knowing a little bit about machine learning, basically these are algorithms that needs to learn from examples. Okay, what are the examples I'm talking about? I'm talking about a query and then a relevant document pair. So such pairs, a query and relevant document, these are kind of the examples we want the algorithm to learn. However, annotating, you know, you can have humans to annotating such pairs, but this is very time consuming and very expensive, because annotating such pairs require human domain knowledge. Okay, you cannot just put in those tasks on the internet and let crowdsourcing to do this. You have to have human domain knowledge to do this. So instead, what we did is to follow the sort of the industry lead, where search engines like like Google, they would take in what has been observed in the search logs. So we know every user coming to PubMed, what the queries they actually entered and what the document they clicked on. Okay, so such information is implicit, but actually give us a good information or good enough information that we can learn or train our model. And that's exactly what we did. So we take a search logs, and of course, we don't look at an individual query. We want to look at a query that are very common and the documents people tend to click by multiple users, and then we aggregate these kind of signals to train our machine learning model. Like I said, the beauty of using machine learning here or learning to rank is that we can generate or we can engineering features. So what are some of the features that we use? So if an article is a review article, that tend to be actually more checked out by our users, so that gets natural boost. If an article is more recent, that gets also boost. Now, even though we do not strictly follow the publication date, but as we all know, researchers want to read more recent papers. That therefore, new papers still gets a boost in that case. If a paper has been accessed widely by different users, says the popularity of the user, says the popularity of the article, that paper also gets boosted as well. So we have about dozens of these different features that altogether, putting them all together, determine whether this paper should be ranked higher than other papers. So all the details, unlike search engines, of the commercial search engines, PubMed, we try to be very transparent about what we do to our users. So all the details are published in this particular paper in the PLOS Biology back in 2018. So here is an example of, I want to show you how this works in action. Now, let's say you did find the paper quite interesting, or the content in my last slide quite interesting, and you want to look for that paper. Okay, now my name are not so easy to remember to you, so that you don't know how to spell my name properly, but you do remember what I'm talking about was best match and PubMed. Okay, so that's the only keyword you need to put in PubMed, and then you do a search. But if you search that by date, sort of order, by most recent, you don't see that paper. However, if we change that to best match, then yes, indeed, the first hit is the paper that you're looking for. Now, of course, I did this because I want to just demonstrate how this is used in action. But when I did this screenshot, I also looked at and found that actually the second paper seems to be very relevant as well about best match. So I did click on this and looked into more details about what this is about. And what I find is that this is a paper or study carried out by external researchers from Canada, and they were comparing best match to the traditional date sort of order, and they find that best match works better, especially for writing large systematic review. So that was very pleased to know as well. Now, of course, this is a very cherry picking because I just want to show you an example how this works. You know, this is not the way that we would evaluate our algorithm. In fact, because every change in PubMed affects millions of our users, when we designed the algorithm, we already did a very comprehensive and very thorough offline evaluation using traditional information retrieval metrics such as precision recall, average precision. But more importantly, we also did something like A/B testing, which is similar to case control study in biology. What I mean here is that we would open up the new feature to 1% of our users, and they will see A version, which is results returned by best match, and B version is results returned by traditional time order. And then we will monitor user actions and see, especially in the click-through rate, whether the results returned in one way or the other mechanism, ranking algorithm gets more clicks. And then we find that best match, in fact, indeed, received a much higher, 20% higher click-through rate. 20% may not be so, impressive to you if you're thinking about perfect is 100. But think about this. When you go to search engine like Google, like a Bing, they have those commercial ads at the very top. If you and that's where their revenue generating pathway or is for users to click on those links. Okay, and if one of those links gets about 1 or 2% click-through rate, that's considered highly successful in the online commercial search engine. So 20% in that context is indeed very impressive. And together, along with the higher click-through rate, we also see a very quick and a very quick intake by our users with this new search algorithm. So we've seen 60% increase in very quickly, in couple months. And now, as you all know, that best match is now considered as the default sort of order used by PubMed, because our users definitely want that. And this is one way of our way of responding to our user need. Now, just to conclude the first story, now if you are someone who are very familiar with this field, you would also know that the learning to rank algorithm is something not so new. It's a machine learning algorithm that was actually first introduced in the 2000s. So it has been already there for 10 or 15 years. Well, there are reasons we chose that, because it's been there and has been so long. It has been very comprehensively validated, both in the research community as well as by industry counterparts. And more importantly, the learning to rank framework is actually very fast. So the production is very different from a research perspective, in the way that we have to have something that can work with hundreds of queries per second. Okay, otherwise it's not just going to able to handle the load of our users. But on the research side, I'm very pleased to say that we are certainly be more interested using the most recent methods, such as those large language models. In this case, we actually have a paper just published in one of the top retrieval conferences later this year, where we look at the relevance of the document to the query, not just based on a query, but based on the semantics. Okay, so the example here is that you can see that the COVID-19 nCoV or SC2, even though they may or may not exist as synonyms in the dictionary, we will automatically can compute the semantic similarity between these words. Okay, so this is one way we call it a dense retriever compared to previous sparse retriever. You have to have the exact term mapping. This way, you can actually imagine that you can have the query, but then very similar document, but may or may not contain the exact same word. And we have shown this actually improves retrievals significantly, and we are now actually investigating ways to incorporate this into the PubMed search. So this is something that we are very excited about. So moving on from our first story to the second step is, now the first step for the first project is really about PubMed search and improving ways that to helping our users to find a set of papers that are relevant to their information need. Okay, and that set of papers sometimes can be quite large, in the number of hundreds, thousands, or tens of thousands. Okay, and we all know that these days, scientists or users are very busy, so there's no way that the scientists can actually spend time or afford time to read hundreds of those papers that are returned by a search engine. Okay, so the million-dollar question here is that, can we teach computers to look through those papers and make connections that otherwise would not be possible by human being? Okay, and to do that, we have to teach computers some basic steps. And the two steps I'm talking about here is, one is, can the computers to understand what is the concept discussed in the paper, and what is the relationship between these concepts discussed in the paper. Okay, so the first problem is, what are things that people most, or what are the most searched topics, or what are the most mentioned concepts in the biomedical literature? Well, it's not surprising, as you can see, that genes, diseases, and the chemicals, drugs, those are mostly often sought concepts by our users, but also at the same time, these are the most important concepts in biological research. Okay, so here is example. I want to show you how exactly what I mean by recognizing concepts from the biomedical literature. On the left side is a free text box. Okay, in this box there's a couple of sentences, and what we want computers to do is to turn and to scan this text, a block of text, and then turn and recognize what are the concepts related concept related, in this case, human phenotypic terms. And more importantly, can we turn and map these free texts into structured ontology identifiers? Okay, so there are two steps. One is to recognize the text that mentioning about particular concept. The second step is map these concepts into controlled vocabulary IDs. So this is known as a concept recognition or named entity recognition problem. Of course, back into our key concepts in the biomedical literature, we want to recognize diseases, we want to recognize mutations, sequence variations, we want to recognize gene proteins, so on and so forth. And this slide just shows you some examples that human language is very rich. For the same concept, there are actually many different ways that a concept can be described. Okay, and this is a very incomplete list. In fact, there are many different ways that diabetes can be described or has been described by the authors in the literature. Human language is also very ambiguous. As you can see that some of the names here these are names in some context, but also oftentimes these are just names to the general English words. Okay, and it'd be ideal, well, if every author stick with the same name for the same gene. Okay, but that's just not possible, because they were saying that biologists would rather share their toothbrush than share a gene name. Okay, I'm not a biologist, but I feel this probably is true because we do see many, many different ways people describe the same protein or same gene in the literature. So this is a problem, and thanks to two very hardworking trainees in my group, we have been working on this problem for the last decade, I will say. And as you can see, we have different tools that have been developed by our group members here, and with fairly good performance. And some of the tools has gone through multiple iterations, like the tool for mutation extraction. We started a project in 2013, 10 years ago, and just last year we released a 3.0 version of the same tool. So these tools are, we are bioinformaticians, so we make all of our tools publicly available because we believe this is one way computational scientists can really make a contribution to our community. And because of that, we find that many of our users, including both NLP specialists or non-NLP specialists, what they do is they download our software in GitHub, and then they just apply these tools to recognize concepts in the entire PubMed, articles. So we thought, if this seems to be a common need for our users, and if every user is doing that, why don't we do this for our users? So this is the idea why we developed a tool called a PubTator, which basically takes the output of all the taggers of different entities, and then which we apply them into the entire set of PubMed articles, PubMed abstract, 35 million articles, and then in the back end we aggregate their results and then present these results in the web browser in the front, to our users. Because I have to mention an important thing is that NLP or text mining I mentioned here is not perfect. So all the tools that we develop here, as you can see, have an accuracy between 80 and 90, which is good, which is very good, compared to other tools, but it's still not perfect, meaning that we would always sometimes make mistakes about annotating something, a name which is not a gene name, for example, or missing some important gene names in the text. But that does not say that imperfect text mining tools are not useful. So with PubTator, we can really show the potential users, what the results look like, and they can judge by themselves whether this level of accuracy is good enough for their purposes. So in addition to the web-based PubTator tool, we also developed API so that people having some computational abilities, they can actually call the API and get in annotation for articles. If in addition to, if they want to download the software, and if people really want to download the entire annotation of the whole PubMed data set, they can go do that through our FTP. So over the years, we've seen quite a bit of usage for our API. We have had now actually close to 900 million or even billion requests since we opened this up back in 2015. And as you can see, our users are from all over the world. So we don't trace our users. We get to know them sometimes through the conferences. They say, they listen to my talk and they say, oh, we are one of your users, and that happens quite a bit. Also, they would cite PubTator so we know what they are working on. So in general, I summarize here, these are the things that we see that people using PubTator. They're using PubTator for NLP tasks. They're using PubTator for assisting speeding up manual data curation. They're using PubTator for bioinformatics. As in the recent years, we've seen people using PubTator for clinical research as well. So one of the things that one of the use cases here, I want to just say one more word is curation, because we actually working very closely with these human curators, and these are the people. Their job is to read full text papers and then extract important information and then put that into knowledge base. And these knowledge base or databases are key for biomedical research these days. Okay, so as you can see, even though they're doing a tremendously important work, but on the other hand, their work is very manual. Okay, their work is not really scalable because you have to read the full papers, and so what we are thinking is, can we help them to improve their productivity? Okay, using AI. And so we worked with several different groups, including the two I showed in this slide. One is a SwitchPod, and the other one is GWAS catalog. Even though they have very different curation scope, in terms of what they would actually annotate or with the biomedical literature. But when we introduce the AI tool into their workload, we find, or working pipeline, we find that MLP can really improve their efficiency significantly, while without any sacrifice in the quality of final annotated project. And that is why they are also very excited and willing to take our system into their production pipeline, because this can really save their time. They can be more efficient and they can spend more time annotating or reading papers. So this is, given the success of PubTator 1, we actually did a step further back in 3 years ago, and 4 years ago now, developed the second version of PubTator, where in the PubTator 1, we only annotate PubMed abstract. Okay, we're using our tools to highlight the genes, diseases, drugs in the literature for the abstract text. In the second version, 2019, we decided, well, even though not all the papers in PubMed, we have its full text available, but we do have a few million of those articles in full text. Why don't we just text and data mine those articles? And that's exactly what we did. So we have some improvement on our taggers, but then the main thing is that we added annotations from the full text, and together we have about 1 billion annotations in the new PubTator 2.0, and that number continues to grow every night, because every day we process new articles coming into PubMed. And that is where PubTator is at this point. So, but in this talk, I would also like to share a good news with you all, that we are actually working very hard on the newer version, the latest version of PubTator. It's 3.0 version. And in this 3.0 version, which is scheduled to be launched later this year, we will not only annotating the genes, diseases, but we also want to annotate the relationship between the gene and the diseases. What do I mean here? Here is an example. You see that there are relationship between the gene and disease and the variant, okay? In this, it's very clear. This is sort of a toy example, because these three things are next to each other in the title. It's very obvious, okay? But this is what we want to do. We want to extract those kind of relationship from free text in the full text articles, and then turn that into structured table or structured knowledge base. But in reality, it's going to be much more difficult than this example, because in an article like this, you would have multiple diseases, multiple genes, multiple variants, okay? Not to mention, sometimes there's negation included in the author statement. So you really have to tease out and find what's the genuine relationship between some of the mentioned entities in the entire article. Now, this particular task is called relation extraction, okay? And it's not a new task. People have done this for at least about 10 years, okay? But the reason I wasn't hesitate to including relation extraction results into PubTator is because the performance isn't so great, okay? So if we look at the field, you'll find that the performance varies between 30% to 80%, okay, 30 to 80, which is a wide range. And that's because it depends really on a particular task and depending on the particular method used in the particular task. So in general, generally speaking, if you design a rules or you're using a patterns to matching, or even sentence concurrence, meaning that if you see two entities appearing in the same sentence, you just assume there's a relationship. These methods tend to perform okay, but not so great. The better method, or the more improved method, is going to be based on machine learning. However, just like any machine learning algorithm that needs training examples, but annotating examples of diseases between disease and genes and drugs is very tricky and very costly. So in the past, there's no data, training data available that captures relations between various kind of entities. There exists corpus, or there exists training data for protein-protein interaction, for disease, for drug-drug interaction. So for one very specific task, but there's no database, or there's no data that would include a relationship between different entities. And that's why we were hesitant to including results in the PubTator. And that's also why we decided, okay, we have to make some data, training data available in order to develop machine learning methods. And that is exactly what we did. Last year, we created a new data set we call it BioRed, which is a large-scale document-level relation extraction data set. It includes about 600 PubMed abstract, and where we annotate, we have 3 annotators, 3 postdoc researchers from my group, and they spend actually almost a whole year to annotate relationships between these 5 different entities. Okay, not only we say there's a relationship between the drug, a disease, and the gene, but we also say what are the particular more fine-grained relationship between these 2 entities, because a drug can actually treat a disease, so that's a treatment relationship, but a drug could also cause a disease as a side effect. So that's 2 different type of relationships that we are talking about. And in addition to that, we actually added another way of looking at disease relations in the sense that, is this relationship that are previously known as a background, or is this a main finding, a new finding of this new article? Because you'll find that the background relation may be repeated over and over again. So we don't want to, sort of adding the known knowledge multiple times. We're really interested is to extract new knowledge from the literature, or the sort of novel relationships. So this is something that we worked with our collaborators at the NCAT, another institute, or NIH, for NLP challenge. And we're going to rerun the challenge this year again, because we now have some new resource committed for annotating additional 400 articles. So total, we would have 1,000 articles in the end, by the end of the year. So with this BioRed corpus, even though it's 600, we are now able to actually develop a deep learning machine learning algorithm that can actually fairly accurately, well, still not perfect, to extract relations between different types of entity types. We can do about 70% or 75% for, just say, there's a relationship between this gene and this disease. If you want to add a fine-grained relationship type, that drops down to about 60%. If you want to say this particular relation type is also novel or background knowledge, that's about 50% accuracy at this point. Now, 50% is not a random here. I want to make sure you understand, because the number of true positives, the number of true instances is much less than the 50%. So obviously, that also calls for more research on this particular task, because we certainly, I believe, that we can further improve these numbers from both the data set perspective and also from the new algorithm development. So this now concludes the second study, which is to extract information from the biomedical literature, from the free text, and then turn that into relations, structured relations in the knowledge base. Now, the both projects 1 and 2 are talking about text. Okay. Now let me switch gear a little bit, and then also show you some of our study that working with clinical images. Okay. This is very exciting, and like I mentioned before, we work mainly in the ophthalmology and radiology, but I'm going to use ophthalmology as example, and in particular, in this case, I'm talking about disease called age-related macular degeneration, and AMD for short. This is the leading cause of blindness worldwide for elderly people, and it's a progressive disease, meaning that prevalence increases with age. And unfortunately, there's no cure for this particular disease. As you can see on the right side of the figure, there's the healthy stage, and then there's early stage, and then late stage. In the late stage, the vision acuity can be severely affected in those patients. But there has been some good news in the case that if zinc has been found the only way to slow down, even though there's no cure for this disease, but high dose zinc can be given to the patient when it's in the early stage or the intermediate stage, can dramatically reduce or slow down the progression. Okay, so the way that AMD is diagnosed by physicians is the physicians to look at the back of the eye images. Those are, we call it, color fundus photographs. And in this case, as you can see, physicians were looking for risk factors, and two main risk factors. One is drusen, the other one is pigment changes, okay, or the color changes. Okay, so drusen are basically those spots on the CFP color fundus photographs in the center, and those are basically fatty drusen droplets deposits in the retina. So there are two versions of the advanced or late AMD. One is the sort of the commonly physicians talking about, and retinal specialists tell me about this, is dry form is a geographic atrophy GA, and then there's the wet form neovascularization. So the challenge here is that even for physicians, general ophthalmologists will not be able to diagnose very well with AMD patients. Okay, you really need a special retinal specialist training for this. And even for them, specialists, this is very intensive and error-prone process. Not to mention, in many areas in the world, there's a lack of access to those specialists. Okay, so it'll be great if we can develop some machine learning or AI tools that can automatically diagnose, or assist physicians to diagnose AMD. And we think we can, because we have some data that are readily available for us to train AI deep learning models. And here I'm talking about two clinical trial data called AREDS1 and AREDS2. These are two, AREDS2 basically is follow-up study with the AREDS1 clinical trial. And AREDS is actually a household name, because if you look at the commercials for the eye drop here, they use the evidence from the AREDS. So in total, these AREDS, they have tens of thousands of images from thousands of patients. And even better is, every image from those patients has been graded, whether this is early AMD, whether this is a healthy control, or whether this is a late AMD. And we have the same patient sometimes followed up and traced for multiple years. So the problem that we want to address then is that, can we develop AI algorithm to severely to detect the severity level of AMD patients? Can we detect the late AMD for our patients, or can we predict, when, at what time the patients will progress from our early stage or late stage, intermediate stage to the late AMD? And this is collaboration with Dr. Chu from the National Eye Institute. So we work together very, very closely with Dr. Chu's team, because these are the physicians. They actually can provide not only the know-how to us, but also give us the actual need in the clinical environment, but also help us to evaluate our AI algorithms that I'll talk about a little bit later. So we developed a tool that we call it DeepCNet. This is a tool, transformer-based deep learning tool that we fine-tune based on the AREDS data that I was talking about. So I'm not getting into the details of a DeepCNet, because you can find this paper in Ophthalmology Journal. But the unique aspect of DeepCNet is, because we work very closely with the clinicians, we can actually model or develop this DeepCNet method very closely with their human thought process, because this would help physicians, if they're going to use DeepCNet in their process, and they would understand how DeepCNet actually makes the decision. So this the ability to explain this process is very important to the users to actually take the AI algorithm. Also, as you can see here on the right is the performance comparing the AI algorithm with the individual ophthalmologists. Okay, so each individual dot is basically a performance of an AI of a physician. And you can see, first of all, physician performance varies, as you can see from this diagram. Some of the physicians do better than others, but the lines here represent the AI performance. As you can see, the perfect system would actually having the performance on the left and the top circle, on the left and top location. Okay, so and then the random is just the diagonal line. But you can see that most of the physicians do work much better, but certainly not everyone. But the AI algorithm actually outperform all the physicians in this particular case. So not only that, you can actually using retinal scans to predict eye diseases. What's even amazing is that we find, or AMD, we can use the same technique for other eye diseases such as cataract, such as glaucoma. But more amazingly is that we find that using the retinal scan, we can actually predict diseases that are from other body part, such as predicting heart attack, so this is a work that we worked with collaborators from the United Kingdom. We also now working on the same idea of using retinal scans to predict cognitive functions, Alzheimer's. Okay, because I guess eyes is really a window to look at the whole body of the health. So ultimately, what we would like to do is, can we actually not only publish papers about these deep learning algorithms, but can we develop tools that can really assist physicians in their in their real world practices? So here there are two questions, right? Can AI assisted tool improve physician accuracy? And can we actually improve also or reduce the time they need for seeing patients? Okay, so for that purpose, we actually developed a prototype, and then we give this prototype and then show the results with two physicians, and then evaluate physicians' accuracy and efficiency with and without AI tool. So we did this with about 20 ophthalmologists from over 10 different institutes. Okay, so they represent a wide variety of different training backgrounds, so on and so forth. And as you can see on the left side, it's accuracy, okay, in terms of both sensitivity and the specificity. So with AI assistance, physicians are consistently perform better than without AI assistance. And on the right side is the time physicians required or needed to making the diagnosis. And with AI, there is almost a 20% decrease in terms of how much time they need for that purpose. So that ends and concludes the third part of my story. And there are many more things that happening in my research group that I don't have time to talk about. Just very quickly, I talked about PubMed, which is the abstract, and I talked about PubTator project, which includes work related to full text. And we have a news tool we call the LitCovid, which actually not only text mine the abstract and full text, but we go one step further to do supplementary materials, because mutations is very highly enriched in supplementary materials. That's why we do this. Last year we had a collaboration work with another institute, Human Genome Research Institute, to look at the literature published on genetic research and to study the use of words related to race, ethnicity, so on and so forth over the last 7 decades. So this just shows you how powerful text mining really is in terms of various kinds of applications. And of course, I I only mentioned about the ophthalmology, but in the area of radiology, we working very closely with one of the scientists and clinicians at the NIH Clinical Center, where we actually not only looking at images or text separately, but we look at them together. You know, because for the same patient, we have their chest X-ray, but we also have their radiology report. So we look at them all together, and then monitoring them to make a better AI algorithm for both diagnosis and prognosis. So what I'm excited about now, as I already mentioned, we will be working very hard, and we'll be launching the new 3.0 PubTator. And the BioRed corpus that I mentioned will be used for NLP challenge. This is through the BioCreative, which is the longest running community-wide challenge event for the NLP in biology. And I was beginning of the talk, I was talking to Dr. Cleveland that, you know, if our talk were about 2 weeks or 3 weeks in from today, I would including our latest research results on ChatGPT and large language models and how they be used for various kinds of tests in biology and biomedicine. Lastly, we are very interested and working on problems to improving fairness in both biomedical search, and in that regard, you know, we want to making sure that every paper has a share fair, every relevant paper have a share fair to be returned, and we want to making sure that users, even when they entering words that are not exactly matched to the words in the document, they can still find the articles of their interest, because we are now seeing more and more, for example, users with their native language as a foreign language. Okay, for example. In terms of disease diagnosis, we want to making sure the AI algorithm that we develop can diagnose patients of different color, of different age group, but with the same consistent results, rather than you do well in one subgroup but not on the others. So with that, this work, I present this work, but it really represents the work from my group, many very smart and hardworking members of my group. Also, I appreciate the opportunity to work with a number of great scientists, both within and outside NIH. With that, I will thank you for your attention, and I'll be happy to take questions now, or you can also feel free to email me later as well.
Well, first of all, Dr. Lu, I want to thank you before we get some questions. I think this has been absolutely a marvelous talk in terms of motivating and inspiring our teams in here as an example of what you all have been able to accomplish. And I think one of the things that is exciting is to show also the journey that you have gone, that you have been building your research on previous knowledge. I do have some questions, but I want to give the audience the opportunity, to ask questions. I know that Dr. Haiwa has some, Shen has some questions on it. Dr. Haiwa, Chen?
Yes.
You mentioned that you have some questions.
Some notes. Yeah. So I have actually two questions. Let's ask the first one. So I'm following your tour called the LitCovid. So like you build a very large data set, you tracking the research articles for COVID-19. So I mean, could you please explain how do you harvest the COVID-19 literature? Because like in that data set, so you only mentioned that you like collect the data related to the disease, the treatment. But actually, in reality, there are many articles talk about the COVID-19, but actually talk about the effect, the social effect, like the online learning, something of the COVID-19. So how do you constructive query and then filter out the articles not relevant to COVID-19 itself?
Yeah. So well, thank you very much for the question. So COVID-19, the LitCovid project started actually before the official announcement or declaration of the COVID back in March 2020. because at that time, like I mentioned, the main driven motivation for us to develop LitCovid is because we know there's a need for scientists or general public to be informed about the latest research about COVID-19. But at the time, if we do a query in PubMed with, well, because you don't know what to search for, because COVID-19 was, the word COVID-19 wasn't there at the time. And if you just do a coronavirus, then you'll get a lot of papers that are about, not about COVID, about other SARS related to diseases. So that is the main motivation we developed LitCovid. And like I said, you know, we were doing this more or less manually at the very beginning, but then we realized quickly this is not gonna, you know, we have a very small team. We, you know, the only person, you know, I had the idea, and then two of my postdocs basically worked on the project over the 2 weeks and to put this together. And then, but then we were scanning the literature in a whole PubMed literature and trying to find relevant papers for COVID. And we did that for a couple of weeks and then realized, you know, the speed of COVID-19 research is really taking off, and many more papers are published. So there's no way we can do this manually. So at that time, we decided we should have to continue to or use our own research expertise in AI and machine learning to identify relevant papers. Okay, and what's nice about this is we have already done this for a couple of weeks. So we have a good set of papers really about COVID. Okay, even though they may not have the word COVID-19. Okay, so that is our seed training data, and then we can then develop a machine learning algorithm that can classify all the new papers that are whether they are relevant or truly about COVID-19 or not. And then, of course, machine learning is not perfect, but we actually have very high probability, high accuracy in the 90s, in an even high 90s. With that said, there are still papers where the machine learning give a confidence score in between 50 and 60. So those are the papers, or between 40 and 60. So those are the papers machine learning are not so sure about. And that's where we still have humans to review and then to making sure they are truly about COVID or not about COVID. And then on top of that, what we did is to annotate important concepts. So or category topics, you know, because COVID research can have a wider variety of different research aspects. They can be about disease mechanism, they can about treatment, they can about transmission, and so on and so forth. So we categorize the COVID papers into, I think, about 7-8 different groups. Some of them are very general topics like treatment. Some of them are a little bit more specific about infectious diseases like transmission, because, you know, and then later in the year, or actually last year, we also added a new topic, which is long COVID, because this is a very COVID specific topic. Okay, but now let me tell you, so the new challenge we have today is that we have many papers in PubMed. They have the word COVID-19, but they are not about COVID research anymore. Because COVID is so common, it's like the word cancer. It's a so common disease now that people just want to mention that for maybe adding the importance of their research. But if we read the research, it's not about COVID-19 research at all. So we now have to sort of to remove that part of the paper from the entire literature, and then extract the ones that are only about COVID research.
Yeah, thank you so much. Very clear explanation. Thank you. So my second question is very simple. So like regarding your second project, when you extract the entities and the relations, do you need to rely on a well-defined ontology? So that can guide you to extract the entities and the relations?
Yeah, great question. Indeed. So yes and no. So yes, in the sense that we want to extract the relations and we want to extract entities, and then we want to map them. So if we extract the disease name, and then if we just extract the disease name, that's good enough. But if you want to using that, compare with other articles, you want the disease name to be normalized, to be linked to a standard way. Okay, so in that regard, we're always using controlled vocabulary or ontology. So let me give you an example. So for diseases, we're using mesh, and for chemicals, we're also using mesh. But then for cell lines, then we're using a different ontology, cell ontology. And for species, we're using NCBI taxonomy. So we do need those ontologies in our methods in order to map the extracted entity into those standard ways.
Yeah, thank you so much.
But in the process of processing, we actually don't using those ontologies. Well, I should say these ontologies also are helpful because they have some relationships between those terms that help us to better extract those entities as well.
Yeah. Thank you so much.
Let's see, we have a question. I know that Dr. Hee-Chin Kim made a note about that he heard Patty Brennan, the director of NLM, talking about the volume of the publications and how hard it was to manage that. And the human-computer collaboration that you're talking about, it would make a difference. Do we have any questions? Help me, Adam, in there. I know that we're getting close to that time. I do want, and we wait. Dr. Kim, thank you for your posting. Dr. Lu, I think of course, we know that it's absolutely marvelous, the work to relate the relationship between a disease and the genes, which is all about right now, and the variants. I was most interested in the AMD because my mother, for personal reason, had it. And if you would know, it's supposed to be, or it's understood that it's a hereditary condition. So what it was exciting when I was reading about your work, it was the fact that not only can make the diagnosis, but it can know the prognosis, which is one of the things that when I go to the eye doctor, it's not always clear, what is there. So let's just say, so my point to you is that I know you're a researcher, your team is a researcher, but we always had the challenge that how do we go from the bench to the bedside? And so to me, I want to know is there any plan? And I know it will not be necessarily your team because you dealt with 10 ophthalmologists, but how is it that this is going to be spread? How are we going to embed that into the healthcare professionals? Do we need to start with the medical schools? And as you know, you look at the curriculum, they're more and more embedding AI. But these cutting edge things, I would think how exciting it would be if my ophthalmologists would have these at their fingerprints, and it could help us to diagnose, and tell me where are we going from here.
Yeah, great. Well, great question. So because I completely, concur with you, because if you look at the literature in the space of AI in medicine, I would say 90, maybe 99% of the papers about a stop at the stage of developing an algorithm and compare with other methods. Okay, only 1% or less than that actually go into be evaluated and be potentially used in practice in the bedside. Okay, so the last mile is really the most challenging part in my view. Okay, so, but like my work in PubMed, what's exciting me about me working at NLM is we get to not only develop the algorithm, but also get to make a difference by those algorithms into production systems. I've been successful with the PubMed so far. We're still working with the condition AI tools, but I can tell you sort of some of the attempts that we have made. One thing I actually already mentioned, and in my talk, is where you develop an algorithm that compares well in paper with the physicians, but in reality, is this something going to be? Because we're not talking about develop something to replace doctors. Yeah. We're talking about develop something as an assistant tool to physicians. So one of the things I already mentioned, we give this DeepCNet tool, and then as an assistant tool to the doctors, and then to see whether or not in reality that can make them more accurate and save their time. So that's one thing we did. The other things I didn't mention, but we have been doing is we also have to evaluate our tool on multiple data sets. We used AREDS data to develop and validate our tool initially, but now we are actually, but AREDS is primarily Caucasians. So we are actually now working with our collaborators in Singapore to validate the same algorithm on Asians. We are going to do that for African Americans and other races as well. So that's another thing to validate, to making sure the tools actually is not biased toward a certain gender or certain race or certain age groups. So that's another thing we have to do before we can really use this in practice. Of course, the last part is how do we engage the FDA in terms of regulatory process. So that's something that I'm least familiar with, but fortunately, my collaborator, Dr. Chu, who's an MD expert, has a great relations with the FDA on this regard. So they have a community-wide meetings discussing the exact things that you're talking about, which is how do we turn a research product into something that can actually be used in clinic. So I think in the next few years, you'll see more and more those algorithms being used and being turned into a product. But this also, well, another challenge I would say is this unlikely to work as a standalone algorithm. It has to paired with a instrument, a medical device. Okay, so by the time physicians using a camera to look at the eye, maybe that results have to be shown directly with the physicians. So these are all this is more like the implementation science issue that all have to be taken care of, investigated before something can be actually used in real time. But I think we're hoping we'll get there soon. Okay.
I can feel it. I can feel it from your answer. We have one more question because you have been so generous with your time. But we have a faculty member, Dr. Hossein Tosamil, and he put in the chat. I don't know if you can see it. If not, I'm going to read it for you. He said, is the sense retrieval model or paper publicly available?
Yeah, yes.
Yeah.
So the one that I talked about is publicly available. The paper is, like I mentioned, it will be presented at the SIGIR conference later this year, but we have a preprint version in the archive. So if you search, it's called LADER Lander, or if you just email me, I can point you to that particular paper. Yes, it is available. Yes.
Sure, I will. Thank you so much.
Great, Dr. Hossein, thank you. I just want to, before I let the floor to Dean Kenchik, who is here now, but Judy Groth put something on the chat, and I think it's a very good way to deal with, because right now AI is getting such of a deal in the news media and society. And of course, there are good points and there are negative points. There are benefits and pitfalls to it. But she mentioned the humanitarian purposes that are behind the AI development. And then we can see very positive applications. So Ms. Gross, thank you for putting that in the chat. So Dean Kenchik, I think it would be very appropriate if you will close the chat. And I mentioned that you were in the Dean's Council. I know you're here.
Certainly, yeah. So thank you, Dr. Lu. Sorry that I was late, as Dr. Cleveland just mentioned. I was in Dean's Council, but I think I at least heard you for sufficient part to get some understanding. And the beauty I saw was that your talk was so accessible that a person like me who doesn't really have much, in knowledge in health informatics area, I was able to follow along. So thank you for doing this, and certainly thank you, Dr. Cleveland, for this series. I don't know if everybody here knows that we also have a Master of Science in Health Informatics program, full program starting from fall 2023, and and the interesting part was that before its website was up, we had to close the admissions because we had too many applications. So that shows the quality that people are expecting based on what Dr. Cleveland is providing leadership for the health informatics concentration in information science masters. So I'm looking forward to that. Thank you for this talk, Dr. Lu, and I really enjoyed it. The part that I attended, and looking at the participants numbers, I'm pretty sure others also did the same way. So thank you both, Dr. Lu and Dr. Cleveland, for providing this opportunity. Thanks.
Thank you, Dean Kenchik. Well, Dr. Lu, I think with this we will conclude. So you can tell you had a wonderful audience. We were over 200, which shows that there's an interest, not only in the literature, but in the clinical side, because your title indicated that. So thank you to you, to NCBI, and to NLM. And we hope in the future to have a collaboration with you even further. We start talking about chat, GPT, and the impact in healthcare and your work. So this might be a good topic for next year.
Yeah,
thank you so much.
Thank you so much. Yeah, and like I mentioned, we have some work already done in that regard. And if someone interested, feel free to send me some email in a couple of weeks, and I can point you to some of our most recent papers on that regard.
Thank you for your generosity. I want to thank everyone who joined us today, and I hope this provided good food for thought and inspiration and motivation to continue to do your work in health informatics, data science, information science in general.